Hello, all. I was thinking maybe you would like if I quickly, and I hope to do it quickly, go over some study guide answers. Um, I know, explain each type and give an example. Mechanoreceptors. Um, so this is something physical. Excellent examples, the ear. Chemoreceptors, we're responding to some sort of chemical molecule. And great example would be taste or smell. Electromagnetic receptors, you're responding to electricity, light, or magnetism. Of course, we have the eye. Thermoreceptors, we're responding to temperature. Interesting. Of course, you're going to be on your skin and stuff, but also you have uh, taste buds that respond to temperature. They respond to capsaicin, um, which you find in hot peppers and jalapenos and all that delicious stuff. Pain receptors, pretty much everywhere. You even have them internally. They're also called nocioreceptors. Then what's the difference between perception or the definition of perception? Perception is all constructed in the brain. So it's formed in brain as your brain responds um, to signals from your senses. So perception is formed in your brain between what you see and what you perceive. Um, and explaining this optical illusion, we know what we see. Our brain is lengthening this. We are perceiving it to look longer based on experience and the way the lines are working. Now, statocysts are the whole cell. You're going to find these in invertebrates. And it is to sense gravity. Gravity. Now the statoliths, like monolithic, large rock, statolith stones, they're what's on the inside. And depending on where the animal is, it's kind of like shaking a ball. If it turns a little, these rocks or these statoliths will fall and start bending the hairs on here. We're talking mechano, but it's a way to respond to gravity. That's how statocysts and statoliths function. Now, your ear structures, I think you all are awesome at the ear so far. Your dominoes went great. Let's see. The sense of equilibrium is centered in the ear. Explain how fluid-filled semicircular canals and otoliths allow you to detect motion. Basically, you have fluid moving over hair cells, moving over hair in, let's say, in a gel, in a fluid moving over hair in gel, bends hair and depolarizes. It's going to cause a depolarization. So mechano, and that's going to be for acceleration. You're in a car, whoo, you're going to push things back. And as I explained, dizziness. What type of receptor is found in taste buds? So receptor, it's a membrane protein receptor. It's a receptor for tastance. 5 types of taste. Remember, we got sweet, sour, bitter, salty, and umami. Have you ever been asked to do taste test and draw a map of your tongue? We know that's all a load now. As with taste, receptors for smell are also integral proteins. But remember, these are integral proteins. These integral proteins are on the actual neurons, not the modified epithelium. This is straight from the book. You should be able to label everything there. Rods, the function, so they're going to detect light and dark. Of course, both of these are on the retina, but this would also be the edges of the fovea. Oh, my pen doesn't write over here. Cones detect color. Oh my gosh, this pen's nuts. It is also on the retina, but they're in the center of the fovea, so you're going to have a whole bunch in the center. Draw a diagram to show the conversion. Okay, so if you have light hitting, remember you have retinol and opsin. Let's put the little molecule in here. And it's when light is hitting it, it's in a straight line, which causes calcium ion channels to open. If it's dark, let's draw the protein. The opsin is bent or in the cis structure, which doesn't cause depolarization. Now, once again, G proteins for the signal transduction use the same way. Skeletal muscle is attached to bone and is striated. What does striated mean? Light and dark bands. Dark bands. Now, what's the next page? This is kind of cute. You're supposed to kind of go through. So, what is a thin filament? Well, that's actin. What is a thick filament? That's myosin. What is a sarcomere? That would be a functional unit. Myofibrils is a bunch of sarcomeres. And what's a muscle fiber? A bunch of myofibrils. And what's a muscle? A bunch of muscle fibers. So just try to remember the order and you'll work yourself all the way up. Below, draw a sketch of a sarcomere in a contracted muscle. On your figure, label a relaxed and a contracted. Okay, so always start with your thick filaments. If it's relaxed, it's going to be longer. So let's make it about this wide. And the actins are the thin ones that go in between. So relaxed is always going to be, come on, pen. Now, contracted, these guys don't move. So you still have your big myosins. But the actins are going to kind of walk closer together, almost touching. So you actually get a shortening of the sarcomere. So total length here is shorter than the total length here. On your figure, label actin and myosin. Actin is the thin, myosin is the thick. Describe the myosin molecule. Okay, so this is a molecule that will change configuration in response to ATP. Basically, the little myosin heads will bind, and we'll get to see that. Oh, excellent. Bioflicks on that too. I recommend you watch. Oh, okay, so here's even more. In the relaxed muscle fiber, the myosin binding sites are blocked by regulatory protein bound to actin. Okay, so this is tropomyosin. This is your actin. This gray string is your tropomyosin. You can barely see them. Little green dots here are your calcium binding sites, and this whole thing is the troponin complex. In a muscle fiber, the specialized endoplasmic reticulum is known as the sarcoplasmic reticulum. What causes the release of calcium? Well, looking at this picture, acetylcholine in the synapse between a nerve cell and a muscle cell. This creates an action potential that actually transverses down this T tubule, and that T tubule causes, this is the sarcoplasmic reticulum, to release that calcium. And let's see, then you have to just label this with all the possible details. So that calcium is going to bind here and here, which will open up these circles, which will allow this myosin head to bind to the actin in a low energy configuration versus a high energy configuration, and you're going to get the shortening or the of the sarcomere, and thus you're going to have muscle contraction. But ultimately, look, you have to pump that as soon as this this depolarization stops, this guy starts actively pumping all the calcium back in to the sarcoplasmic reticulum. But you can just copy that directly from your book. Is there more? Okay, video's getting too long. Oh, steps of muscle contraction. How hard can this be? Let's see. The first one is we need an action potential. So acetylcholine, no. Acetylcholine, no. Depolarization continues across the sarcolemma down the trend, no. Nerve impulses arise. So that would be number one. Nerve impulses arrive. When the nerve impulses arrive, we release acetylcholine. Acetylcholine binds to the muscle fiber next. This allows sodium ions to rush in. So that depolarization causes the transverse tubule system, which is going to cause the calcium to be released from the sarcoplasmic reticulum. Well, what does the calcium then do? It's going to bind to troponin molecules, yes. Troponin-myosin complex is moved, binding site, the actin is exposed, yeah, 7. Myosin head rotates, yep. 8, in the presence of calcium, myosin acts as catalyzes the breakdown of ATP. Energy is transferred to myosin and myosin. Okay, so I got this one wrong somewhere. Let's see. Calcium ions bind to troponin molecules of thin filaments in the presence of calcium myosin. You can figure it out. My video is too long. Well, we can also go over that in class. Holy Hannah. Okay, this is a long one. I'll make another recording and finish it up. Stop.